Hello everyone, my name is Andreas Falk from BioNanoNet. I want to warmly welcome you to the BioNanoNet Member Welcome Webinar, which is already the 5th in the series. And we just immediately start with some housekeeping rules. You know it already since some years now, that please use the question function if you have any questions. Questions will be asked then by the moderator based on the entries in the chat, so that we have a smooth run of the session. And please note, this webinar will be recorded and you will have later on the opportunity to see it on the YouTube channel and have later on digest of the presentations of our new members. Our new members are not alone. The BioNanoNet Association has already more than 70 members, and we are really very happy about this nice group and community, which is more and more growing, and where you can see also the developments of this group in the BioNanoNet Quarterly, which is coming out every 3 months, and it's named our digital magazine for developments in sustainable technology. Here you have a QR code, so you can immediately scan it and register for it if you're not yet subscribed to receive it. With this, to not further delay the presentations, I want to give you the agenda. We will start with Carlos Sánchez Somolinos from Advanced Manufacturing Laboratory, and then go to Covirabio, Marko Poglitsch. So now I'm happy to shortly introduce Carlos Sánchez Somolinos. We are collaborating already in a project called Prime. He is in Zaragoza located. He will tell you quite very well about Advanced Manufacturing Laboratory, which is more than one group. And we are really very happy to have you on board of our network. And I'm quite sure that people will learn a lot from your presentation, what they can get from your end. With this, I'd like to hand over the screen to Carlos.
Thank you very much. Good morning. Thank you very much for your kind introduction, Andreas. I would like to thank you and also the BNN Association for the warm welcome and for giving us the chance today to present our activities to this community. Okay, we go into business. This is the group of people that make things happen. Here on the left, we have Elena and Maria supporting the administrative and technical tasks in the group. And on the right, we have Lorena Ciamanos, Mersat, Lorena Montesino, Eric, Paolo, and Augusto that push the research ahead. I must say that I'm very proud of all of them. We are all CSIC employees. For those of you not knowing the CSIC, this is the Spanish National Council of Research, and it's the largest public research organization in Spain and the 3rd largest in Europe. Some figures about the CSIC: it has 13,000 employees working in 120 institutes that develop research in 8 quite different areas of research. And one of these areas, material science and technology, it's where our institute, the Institute of Nanoscience and Materials of Aragon, the INMA, is developing activities. By the way, we are located, as Andreas mentioned, in the beautiful and old city of Zaragoza. I kindly invite you, I kindly urge you to come and visit us. Well, within the Institute, at the Advanced Manufacturing Laboratory, the group that we are introducing here today to the BNN community, we focus on the design and preparation of structured polymeric systems with functionalities of interest in optics, in biomedicine, and in soft robotics. More in detail, we focus on functional organic materials, mainly photopolymers and liquid crystals, and sometimes incorporating functional molecules or functional nano or microparticles that provide extra functionality into the material. Okay, to turn these materials into useful systems, into functional polymeric systems, we need to control their morphology, and we need to do this at different length scales, from nano to macro, to the large scale. And for this, we rely on a toolbox of manufacturing techniques, of advanced manufacturing tools that includes self-assembly, that is the capability of matter to organize itself at the molecular level. We also count with photolithographies, direct laser writing, as well as other additive manufacturing techniques like inkjet printing or 3D printing, in which we have nowadays currently a special focus, as it will be clear in the next slides. Okay, with all this, we carry out research in different areas, as I mentioned: optics, generating microstructure for refractive, diffractive, and polarization optics; biomedical applications, producing hydrogels with tunable properties, for example, tunable different mechanical properties; or microstructures, trying in all the cases to generate biomimetic microenvironments for cells. And finally, we also, as mentioned, work on soft robotics, in which we are bringing together new materials and new manufacturing technologies, methodologies, to create new opportunities in areas that cover, for example, as we will see, active microfluidics or other biomedical applications. I want to focus in this last topic, soft robotics, in which we are especially putting our efforts nowadays. And to start with, I introduce the concept of soft actuators that you have there. These soft actuators are soft polymeric systems that deform in a prescribed fashion in response to an external stimuli, for example, heat, light, or an electric or a magnetic field. Okay, these soft actuators, in contrast to their hard conventional counterparts, as we see below, these soft actuators are characterized by presenting flexibility and adaptability in the deformation that allow them to handle delicate objects, as shown in the picture down right of the screen. Well, due to these characteristics, these soft actuators are interesting, for example, in the implementation of robotic functions with potential use in minimally invasive surgical devices, as they can handle delicate tissues. Potentially, they could handle delicate tissues without damaging these tissues. Okay, and in this field of soft actuators, we focus on this family of materials, liquid crystalline polymeric networks, that are composed by this. You cannot see the pointer, but it doesn't matter. These elongated rod-like molecules that are incorporated into the polymeric network, as shown in the cartoon in the upper right part of the slide. Importantly, these materials present an isotropic deformation when they are exposed to an external stimulus, for example, an increase in temperature. And why does this happen? Well, this increase of temperature produce disorder, molecular disorder in the system, and as a result, these elongated rods get disordered, get more oblique angles with respect to the preferential orientation direction. And as a result, and due to the linkage of these molecules to the polymeric network, this disorder induces a contraction of the material in the preferential orientation direction and an expansion in the orthogonal directions, as we see in the bottom part of the slide. And this is a key aspect that it will appear more times now. Okay, using this principle of deformation and controlling the morphology that is the orientation of the molecules, we can implement controlled deformation. And as a typical example that is ubiquitous in the literature, here you have a cantilever in which the molecules lie parallel to the surfaces of the cantilever, but their orientation is orthogonal in one side with respect to the other side. So if we hit this cantilever, the material, as mentioned in the previous slide, try to contract in the preferential direction of the molecule. So the upper part of the cantilever try to contract in the direction indicated in the slide, in the cartoon. But in the bottom part of the cantilever, due to the orientation of the molecules, the system is try to expand in this direction, and the system, to accommodate these stresses, goes out of the plane and bends upwards. Okay, this type of effect and this type of configuration has been used by different groups and is reported extensively in the literature to demonstrate various actuators and devices useful to make grippers or microfluidic mixers or even walking devices, as we can see down right in the slide. However, the fabrication techniques, the traditional fabrication techniques that are adopted from the liquid crystal display industry, limit to thin films of these materials, that they have limited capacity, limited mechanical performance, and also the patterns that we can get are very limited. So we are limited in the development of this technology. Yeah, in this context, in this scenario, we have developed a printing platform of liquid crystal elastomers. We did this at the same time and independently from two groups in the US, the group of Professor Taylor Ware in Texas and Jennifer Lewis and Timothy White in Harvard. That essentially they demonstrated simultaneously the similar principle. In our lab, what we have done, we have prepared this the main chain polymer that you see over there in the top part of the slide, with having a reactive acrylate groups, and this polymer chains together with a photoinitiator, a molecule that allows the polymerization of the system. We have printed these chains, and later we have fixed. After the printing, we have fixed the material with UV light. The UV light excites the photoinitiator, and it polymerizes this acrylate containing molecules. We can iterate this, and we can generate several layers. We are using 3D printing, so we can do these things, and we can therefore grow into the third dimension. Here you see lines of materials printed at different speeds, so they have different cross section. And what we saw advantageously for us is that, especially for high speed movement of the nozzle of the needle that dispenses the fluid, that dispenses the polymer, for high speeds, and this is a key point I want to call the attention, because this is a key point for this technology, we get a preferential orientation of the molecules aligned parallel to the direction of movement of the needle, as you see in the cartoon bottom right part of the slide. And this is the key point, because we can, with this printing platform, with this material, we can digitally control the orientation of the molecules in the printed structure. Okay. Okay, now we get 3D printed elements, and we want to see how they evolve in time when we expose them to a stimulus. I see we want to see how they evolve in time. Time is the fourth dimension of 4D printing. When we stimulate, the material changes the shape, so it evolves in time, and this is the fourth dimension of this 4D printed platform. And here we have a continuous layer consistent of parallel adjacent lines of polymer, with the molecules aligned parallel to the long side of our element. Okay, when we hit this element, and the molecules get disordered, as we described before, the materials contracts along the direction of the molecules and expands in the perpendicular direction, as it is expected in elastomers, as we saw a few slides ago. So key also in this technology, the digital control that we have achieved in the molecular orientation with our printing platform allows us to digitally control the stresses within the material when we hit the material, when we expose it to the material to an external stimulus. And this is what has allowed us to have access to complex deformation modes such as these emerging cones that you see above, or multi-layer artificial muscles that can rise heavy objects, which we overcome therefore the limitation that we mentioned of conventional processing methodologies. And we also have generated in the bottom right part hybrid elements integrating liquid crystal elastomers. You have there a yellow ring of elastomer, of liquid crystal elastomer, that is able to deform a transparent silicon PDMS layer that therefore, as a result, form a lens with a focal length that depends on the temperature. So we can modify also other materials with our technology. We can also generate other complex elements like this one you see there. It's made of one single material that is capable to perform multiple tasks, like a couple of robotic functions, gripping an object and traction, raising the object in response to heat. We are just heating the element and we see these complex functions working together. We can digitally embed these functions in the material with our materials and with our printing platform. Okay, based on this 4D printing technology, we coordinate the FET Open project PRIME that already was mentioned. This is the consortium, including our colleagues from BNN, Andreas, Kaitlin, Simon, Barbara, and all our colleagues from BNN. And well, in PRIME, our ambition is to use this 4D printing technology, these materials, to implement fluidic, all the fluidic functions of a microfluidic chip, that is the valves, the pumps, to integrate them with our materials in the chip itself. Our objective is that the elements can be also remotely actuated by light. This was our ambition. Eliminating therefore the need of external equipment, external pumps or external valves. We want to integrate all the materials with this 4D printing of LCEs, with the liquid crystal elastomers. Well, I can say at this stage of the project that the consortium has successfully incorporated photoresponsive molecules. We have very nice materials responding to light, and we have also successfully, very glad to say, we have implemented active microfluidic valves that can regulate the flow of liquid in response to light. Also in this swap robotic topic, we coordinate this Maria Sklodowska-Curie network of excellence StoreBots. And in this case, we are 9 beneficiaries, 9 partner organizations, academic and non-academic. And here our objective is a bit different. It's not only to develop technology, but it's the main target of the project is to train these 13 early stage researchers, our ESRs, in the area of liquid crystal elastomer-based actuators and soft robotic systems. In particular, our ESRs, Eric and Paolo, and working on developing, and actually they have succeeded on developing materials, very nice materials that are printable and they respond to light and also to magnetic fields. And we are going to explore together with other partners their integration in surgical devices. Okay, beyond this European project, we are also exploring the frontiers of additive manufacturing with this type of materials. And here I would like to highlight our recent work on melt electrowriting. and just say that with this technique we have accomplished the digital generation of active structures with unprecedented, I can say, fiber sizes, very thin, ultra fine fibers, and very small periods that you see in the same images, opening in this way new opportunities in the field of smart, responsive scaffolds. I end here highlighting our reasons to join BNN and also our expectations. I want to say that, as Andreas mentioned, I met Andreas 4 or 5 years ago, and I have to say that BNN has been a very reliable project partner. So we look forward to future joint enterprises, to future joint projects together. And yeah, we will also like to interact with other BNN members and of course become a key partner in the association in the topic of functional polymeric materials, this type of smart materials. And to finish, it is our ambition to see our materials, our smart materials, and also our software robotic functions, our implemented functions, being used in relevant applications, in relevant technologies. And we find BNN as an opportunity to accomplish this goal. This is the last slide. Now this is already the last slide. I just would like to thank the institutions, the funding agencies, of course, that facilitate our work. Again, Andreas and BNN for the kind invitation to this present welcome webinar, and of course all of you for your kind attention today. Thank you very much.
Thank you very much, Carlos, for your great presentation. I would say this gives also a good appetizer what the people can get from not only your group, but the groups you are representing here in this network. One question is pending in case of the application orientation. So which applications would you say are next door for you already as, in your developments? Which fields, medical field or other fields, you see the, let's say, the closest next step for your developments?
Yeah, thank you, Andreas. Yeah, indeed, as you well know from Prime, from our project on active microfluidics, we really would like to expand this. We have already done proof of principle of this light actuated valves for microfluidics, and we would like to bring this technology to a more mature TRL. We are now in a TRL 3 or 4. One of the things, as you well know, is we would like to go further, and we would like to push this ahead. Also, as I mentioned, we would like to explore the use of this system, especially the magnetic ones, that we can have a very good three-dimensional control of these systems in the use in their integration in surgical devices. I mean, we have one of our partners in one of the projects is a robotic company for surgery, and we are exploring possibilities to integrate these materials and to see the potential of these materials in this type of thing. And one other thing, the final application that I would like to mention is this scaffolds, these active scaffolds. Typically, you have a scaffold, and typically these are static structures. You have a microstructure that hosts cells, and but typically these are static elements. We aim to make these scaffolds active, so we can more biomimetically emulate the environment of the cells. I mean, cells in the body are not static; they are stressed periodically, sometimes in the heart, in the gut. So we would like to also to emulate this dynamic behavior. But this is a future thing.
Thank you very much, Carlos. This is also a perfect link to our next talk. Carlos, thank you once more for a nice presentation. We will now switch to Covirabio. Marko Poglitsch, one of the co-founders of this nice organization, is today our presenter. I would say you are in the field of revolutionizing diagnostic field to a certain extent. It's great to have an Austrian organization, SME in that case, which is very relevant, of course, also then in research projects to be partnered there, but also as a success story in the IVD field. So I want to give you the floor, Mr. Poglitsch. We are really very much interested in your presentation and to learn also. I think the audience should hear where are the connecting points to collaborate with you in the near future.
It's a great pleasure for me here today, and to thank the organizers for the opportunity to present our company here to this network today. I was thinking I will give you a brief journey into the beginning of Covirabio, because at the time where we started, which was the beginning of 2020, I think all of us encountered a certain challenge. So the pandemic kicked in, and when historically driven, with my position as the CEO at Adderquant Diagnostics at this time, we were very close to the biochemistry of this coronavirus, keeping us all busy at this time of the year. So when the pandemic started, it became soon clear that the entry receptor of the coronavirus is recombinant, is angiotensin converting enzyme 2, which is an enzyme that is part of the renin-angiotensin system. And with our diagnostic services that we employed at Adderquant, and our knowledge from our involvement in clinical studies, Peron Biologics, so another Vienna-based biotech here that might be familiar to many of you. We had quite some knowledge about recombinant human ACE2 at this time, and also the ACE2 molecule and its structure and functions. And so I remember a phone call very well where I talked to Hans Leubner, the former CEO of Peron Biologics, and we were thinking, how could we use this knowledge, this molecular structure of ACE2 and its interaction with this coronavirus leading to this high infectivity, how could we make use of this feature diagnostically? And so we, at the same time, there was a lot of trouble with the PCRs that were rapidly developed at this time to diagnose the coronavirus. And so I want to put up this slide saying quarantine and innovation, and what does it mean? So at this time, being positive for a coronavirus was quite a problem, because the quarantine was very long at the beginning of the pandemic, and on top of that, patients being positive, PCR-based positive for coronavirus, stayed positive for a long time. And this slide should illustrate how this could be explained. So basically, when you take a swab of a sample in the throat or mucus, the cells that are infected by the virus containing multiple components are disrupted, and the resulting sample is a very, let's say, biochemically dirty mixture of components containing epithelial cells, cell fragments, virus proteins, free genomic viral DNA, microvesicles or micromembrane structures, intracellular RNAs, and also infectious virus particles. And if you do a PCR, a real-time PCR, out of such a mixture of components, we cannot really tell where our signal comes from. So it could be that we see patients that actually are highly positive for coronavirus, but they are not infectious anymore. And this happened to a lot of patients being in intensive care units that could not be discharged and still being positive for the virus. In fact, they weren't infectious anymore, which could not be detected at this time. And we thought, okay, if we would be able to process the sample in a way that we select infectious virus particles based on the natural virus receptor, it should be possible to generate a much more specific signal for infectious virus particles, and that is the now we call it the VERB assay, virus entry receptor binding assay, which utilizes the interaction between the natural receptor for the virus that is interacting with the trimeric spike protein on the virus particles. So the VERB approach basically mimics the natural infection events, which is depicted here on the left side. So the virus attaches to the receptor, then there's a complex machinery ongoing that involves membrane fusion, entry of the viral RNA into the cell, involving several accessory molecules, and then replication of the virus inside the cell. And by doing preceding the VERB step, the enrichment step, to the RT-PCR that can be used to detect this kind of particles, we get this high specificity for infectious particles. So the sample material is swabs or body fluids, but meanwhile you can do also cell culture and other biological samples. The technology is compatible with different detection methods like colorimetric or fluorimetric approaches, real-time PCR I already mentioned, of course, surface plasmon resonance, biosensors, and also microfluidics. And we have done a proof of concept study in COVID-19 patients together with hospitals here in Vienna. So we analyzed 214 samples from COVID-19 patients and compared the VERB PCR with classical PCRs, did correlations. But as you can imagine, all these processes take quite a while. And if the pandemic has taught us something, then if there's a strong pressure on society, then development can be really fast and efficient. So the PCR development that we have seen for COVID-19 was really impressive, and soon the scale up and also the improvement of speed and technology in PCR-based method allowed to screen patients or screen potentially infected patients on a broad scale. So that's the status now for the VERB assay for virus detection. We developed a kit that is applied in research right now. It can be used for isolation of intact virus particles from patient samples. So it enriches the viral particles, can be combined with all the techniques that I mentioned. And secondly, this kit can also be used to detect and quantify neutralizing antibodies in samples, where again, this interaction between the natural receptor and the natural entry molecule of the virus spike protein is employed and analyzed through appropriate methods. So that was a little bit of a journey back, and if we want to summarize the challenges in the diagnosis of infectious diseases, which let's say a lot of people in the last 3 years have learned the hard way. So the infectivity is a big problem, so that's how it all started for Covirabio. The turnaround time is, of course, important when we talk about highly infectious and also dangerous diseases like SARS-CoV-2. Sensitivity is another problem. There was a lot of discussion on where should we put the cutoff for the PCR. Is the PCR even sensitive enough to tell us whether a patient is infectious or not? Specificity I already mentioned, so it's not very helpful to have a lot of false positive results. Therefore, PCR is a good technology for that, in fact. But as discussed previously, if we get signals from non-infectious RNA, that also doesn't help, and we might end up sending a lot of patients into quarantine that don't have to be there. But of course, also sample characteristics play a role, how complex the sample is. Mucus is not a very easy thing to handle, and yeah, also the accessibility. So where can we test? How fast can we test? Do we need sample logistics to get the sample to the place where it's finally analyzed? So with all these factors in mind and our experience from working on this verb assay for detection of coronavirus, we took some further steps with Covirabio in taking the concept of using host pathogen interactions for diagnostics further. So, and our mission is to identify specific and immune relevant host pathogen interactions, also develop novel and highly sensitive nanoparticle-based diagnostic tools, and we want to implement these emerging technologies to develop improved and affordable diagnostic solution for detection of such pathogens and also for immune monitoring. And we're especially thinking about microfluidics and biosensors in this case, and also to provide these technologies in a close to patient setting, meaning at the point of care. So of course the pre-analysis needs to be robust and ideally also simple, and the whole process should also be rapid and sensitive. So that's quite a high bar, I would say, and the areas that we are currently working in all combined by one common feature. So the platform technology that we are working on are functionalized nanoparticles that are applied in different technologies. So for the verb assay, we use these nanoparticles for sample concentration and also respiratory virus detection. At the same the interaction, of course, is affected by the presence of molecules that are inhibitory to this interaction, so especially neutralizing antibodies. Now, like 2.5 or 3 years into the pandemic, almost everybody is vaccinated, but now the question arises, okay, am I actually protected from coronaviruses or from SARS-CoV-2? And if yes, which is the subtype that I'm protected from? And this technology is also suitable to get this information by multiplexing approaches. So we could look at, by using different spike proteins, spike variants, combining them with the ACE2 as the receptor here, we can get information on subtype specific immunity. So, and the last pillar that we are working on, we call this the phrase assay. It's a phage-based receptor enhancement strategy, and phages. I will get to this a few slides later. But what all these approaches have in common is that they're possible to be combined with a variety of different readout technologies, including microfluidics, PCR-based techniques, laboratory-based techniques like ELISAs, or also can be used for concentrating and purifying pathogens to be detected in the following step. I would like to talk a little bit more about the immunity monitoring aspects, where you also use SARS-CoV-2 as a model for nanoparticle-based detection. So we have different approaches where we follow this strategy, which is on the one hand a paper microfluidic-based approach, and we have a very fruitful collaboration with Peter Ertl here from the Technical University of Vienna. I think he's also part of the network here. Also cartridge-based microfluidic assays, which are developed also in collaboration with the Technical University of Vienna and a group of Professor Ertl, where the idea is to develop a multiplex readout for different neutralizing antibodies. And also we have a lab-based verb neutralizing antibody kit that I already mentioned, that we already developed as a ready-to-use product for applications in research. So how does the immunity monitoring work? It's the same story. Instead of epithelial cells that we see in humans, that are the receptors for the cell types that are targeted by the viruses with their ACE2 molecule on the surface, we are mimicking these by nanoparticles, also carrying the functional, fully correctly folded receptor on the surface. So there's a little bit of a turn to be done here. So the molecule itself, the ACE2, is a dimer. It's a membrane. It's a membrane-anchored protein. So you need directed orientation, dimeric folding. It has some ions in there, which also support the correct folding and function. And on the other hand, you have the spike protein, which is also a highly complex molecule. Both of the molecules are highly glycosylated, and the idea is to use the molecules in as close as natural state, which implies special challenges in the handling of these compounds. So this is an example of the data that we received from immunity monitoring experiments, where we used reference sera for SARS-CoV-2 after immunization, of course, and we diluted this reference sera containing neutralizing antibodies in the verb-based immunity monitoring kit. And as you can see, you can get a very nice dose response curve here for detection of your neutralizing antibodies in your sample, and this principle is then applied to nanoparticle-based detection in the form of lateral flow setups. And this is some examples here where we just can see the control line here. It's the prototype of this test, the control line, and on comparison here the test line, which is showing us a graduated decrease with increasing amounts of neutralizing antibodies in the samples. So the assay is compatible. It's a rapid test. It takes like 15 minutes. It's compatible with whole blood. So there's a blood separation membrane at the beginning of the strip. Yeah, it's based on the natural receptor ligand pairs, which we think is pretty unique to this kind of assays, because most of them are using like short stretches of the spike protein. In our case, we used the fully functional trimeric spike protein to set up this assay, giving additional binding sites for potentially neutralizing antibodies. So last but not least, I want to tell you a little bit about the phrase approach, which I believe is a very promising project within Covirabio. So we all know that infectious diseases or bacterial infectious diseases are a big health problem, especially if we think about urinary tract infection, but there are plenty of others where this principle could be applied. It's again nanoparticle based, and we again use the strong and selective interaction of natural ligands or natural pathogen molecules with cellular molecules. And what we do in this approach is that we employ the, let's say, power of evolution to improve diagnostic specificity. So phages are viruses that are specific for bacteria, and almost every bacterial strain has a specific phage that infects only this bacterium. And this specificity, we make use of this specificity by employing this phage proteins in the phage receptor-assisted sample enrichment to get bacteria purified for further diagnostic workup. And this further diagnostic workup we are pursuing by a microfluidic approach, which also allows not only allows qualitative determination of this pathogen, but also allows for CFU, colony-forming unit determination, and also antibiograms. And yeah, of course, the vision is to have a rapid and fast test that allows us to check for variety of pathogens the sensitivity for individual antibiotics, to give the doctor a rapid handle to treat the patient properly with the right antibiotics, especially thinking about the increasing incidence of resistance against antibiotics by bacterial strains, and just thinking about MRSA in this context. We believe there is big potential in this technology. Finally, I would like to thank our R&D team here. So we are currently 8 people at Covirabio. they are doing a great job, and we have a different areas. We have our own protein production facility. We have our own biosensor facility, and yeah, of course, I would also like to thank Professor Peter Erdl for his support, and also his whole group, who are doing a great job in doing the microfluidic parts for us. And yeah, also we would like to thank our partners at the universities, and also Medical University of Vienna, Klinik Donaustadt, who supported us in the beginning with the sample supply, and also Atacon Diagnostics with experience in ACE2, and also supporting us with lab infrastructure in the beginning. Thank you.
Thank you very much, Mr. Poglitsch. That was very much insight into the immuno activities and all this. There is the one question, of course, how, let's say, near to the market do you see your microfluidic parts already? You were talking about different things, but the microfluidics is more or less the bracket around all this interaction here also in the network. So how far or how close you are already with this at the market?
Yeah, well, I thought I would not show any data, because the idea was to give you a broad overview about our activities here, and yeah, the prototype actually is working, especially for the phrase microfluidics. We still have to combine so the idea is to combine everything to one device that is then used at the point of care, and we still need to combine that with several accessory features, but the basic principle of the microfluidic park is already functional, and now it's about getting the stuff around, getting the regulatory framework fixed, and also getting proof of concept studies on the way. Because of course, if you're implementing a medical diagnostic or a clinical diagnostic device, you have to do your clinical performance testing and your validation studies, and that's to be done now according to the IVDR, which is a big challenge, as many of you might know.
Very, very good point. There might be one interesting thing which should be mentioned here as well. There is a Open Innovation Test Bed. It's a project funded by the Commission, which runs so-called open calls in the microfluidic area. It's called Next Gen Microfluidics Project. Maybe you know it already. We just have put the link for the next open calls, or actually open calls, into the chat. In principle, such kind of research projects, which kind of research projects are the most interesting for your organization?
So we're still, as I mentioned previously, we use the COVID and the SARS-CoV-2 ACE2 interaction as a kind of proof of concept for spreading out on different technologies and see where can we go with this principle. But there are plenty of other applications in diagnostics. So if you just think about parasites, like mosquito diseases, or like ticks, so this kind of viruses that are now spreading out over Europe with the climate change and these developments, it will be definitely a demand to get a diagnostic of different pathogens. So what we are working on is extending the technology to different pathogens that are of big health impact or health interest.
Okay, thank you very much for now. Carlos, may I also ask you to switch on your camera as well for a final round for discussion. Of course, if you have a question to each other at the moment already, feel free to step up and ask this question. If otherwise, I want to have this one connecting point, which is already there in the PRIME project. Of course, Peter Erdl is one of the monitors. in Covirabio. Peter Erdl is one of the mentors, so-called, and cooperation partners in both. The microfluidics is the common bracket where we will meet each other, hopefully also on the 14th of September then in Vienna in a physical meeting. I was really wondering about to have a kind of message from your sides in the microfluidics field. What developments you see should be run in the next sessions in research projects from your end, where you want to collaborate with other organizations. This, Mr. Poglitsch, I just asked you this question already to a certain extent. This is why I would ask Carlos maybe to give a short statement now on this, and then Mr. Poglitsch, maybe you can add some more.
Sorry, the microphone was off. As I mentioned, we are a group more focusing not in the biological part, as you saw, more in the materials and active materials part. Then it's our aim to integrate these materials in and make microfluidic devices different to what we have now. I mean, eliminate complicated pumps and valves from what is now the paradigm in microfluidics. We have made first steps. It's a long way, I would say, that we need to walk. But yeah, indeed, bringing this technology further could allow us to integrate in other microfluidic platforms that are going on in the world, and more specifically in the association. So I think, let's say, we are generating technology that could be used in the future to make chips, microfluidic chips, different. And we are working on this. I mean, we are working on making, elevating the TRL of our technology further. Yeah, this is our goal.
Thank you very much, Mr. Poglitsch.
Yeah, I was with greatest, I was looking at your presentation, Carlos. So I've never seen like soft robotics before, but I think this temperature sensitivity and novel materials is definitely a opportunity for microfluidics, and I think there's a lot of application, especially in the let's say biodiagnostic area, because we do this kind of particles we're looking at. So certain surface, they have surface molecules, certain features, interactions with the pardon, with the channel walls. We sometimes need temperature incubation of the chips. We care about gas permeability, because when we want to have like cells growing in there, we don't want to have the medium, let's say, contaminated by permeating gases or fluctuations in temperature. And I was just thinking right now when the soft robotics, so I was thinking, well, maybe temperature sensitive valves or something could be an application for microfluidics, that because when we take our slide, we put us in we take them to 37 degrees to incubate, to stimulate bacterial growth. So we need switches in the fluidic path. So I definitely see some room for novel developments here.
That's great to hear.
Thank you very much for that. In terms of course, all these applications and all these materials which you mentioned here, okay, advanced materials and smart materials was already mentioned. I think there is also a really good stake you can give towards all the sustainability efforts underway at the moment. So maybe you can, let's say, conclude our short webinar with some short statement also, where you see the highest impact of your research towards this getting more sustainable with the products, with the technologies. Let's say, as you said already, with one material use instead of a complex combination, maybe all these elements, how far you would go, how much impact can you gain also with your research towards getting all this microfluidics field more sustainable?
Well, I think it's not just about research. The research is the one thing. We're the experts in our fields, and we know how to develop, say, diagnostic assays and to validate assays. We know about the pathogens and about the importance for human health. Carlos, you are the expert in doing materials, 3D print, and we're doing research in this field. But also we have to think about how can we get this to the market and how can we commercialize that. And from the diagnostic perspective, we have some room for with the nanoparticle-based technology that we are using, but by combining two research and combining two innovative approaches to solve a problem, we reach a new level of innovation and also patented. And as soon as we have patented, we also get the chance to get investors to develop products to bring them on the market, and without bringing market, it will not help anybody. I think the goal that we're aiming for is also IP and yeah, getting translated to the market, getting products efficiently.
Thank you. A very valid point, and I think Carlos is also well approaching also to the market with his ambitions. Carlos, please, you have the final words now.
Yeah, you mentioned one very important word, sustainability, and the materials that I mean, we many times work in the lab, and we don't carefully look sometimes to the life cycle of the materials. When, as Marko said, you think in a product, you need to think that this device that you're generating, it will be hopefully generated in millions of units or many units. So their sustainability and life cycle of the materials is very important. And I'm glad also to be able to say that we, in our project in Prime, we have also taken care of this, and these materials, these elastomers that initially in their initial conception, they were elastomers, physical elastomers, that they are not easily recyclable. One of our partners has come with a concept, a new synthetic concept. Technical University of Eindhoven, Professor Senin has come with a concept of new responsive materials, printable materials that can be recycled. They are thermoplastic based. So we also need to take care of these things, even if the piece of material is very small in the system. We really need to take care of every detail to make everything more sustainable.
Definitely. Thank you very much also for this statement, and that you already integrate in your research work the thinking about the future, the thinking about sustainability. I think that's also one of the core items we try to push forward also in our community here. As you know, as the BNN also runs a lot of things in the safe and sustainable by design field. For this now, for the webinar, I want to thank you very much once more for your great presentations, really very interesting stuff. We will put them, of course, on the YouTube channel, and to be able to share it also with your partners, with your interested groups. Let me give two more, let's say, save the dates. On the 31st of May, there will be one part, a sustainability webinar in the railway sector, which might be not the direct connection with your business. And the second part on that day will be from the Austrian NanoNet, which is located in Vienna. They will run in our project NanoSyn a workshop with really interesting talks, but more related, of course, to other materials which are really interesting also, most of them in the nano field. And the second what I want to mention is September 13, Vienna. We hope to see you all at our annual forum. As far as I know, Carlos will be there in physical form. Hopefully, Mr. Poglitsch can also join, or other colleagues from Covirabio as well. We will have a core topic which is around biofunctionalization, which might be quite for both of you already a very interesting topic, but also for others to join. And it will be in Vienna, which seems easier to reach for the most. With this, once more, thank you very much, Mr. Poglitsch. Thank you very much, Carlos, for your great presentations. And I want to also thank to my team, Babsi, Simone, Caitlin, Beatrice from the communication part of BNN. They did a great job in setting up this webinar again. And yeah, with this, thanks also to the audience for joining this meeting. And I'd like to close it now and say goodbye and wish you a nice day. Thank you very much.
Thank you.
Thank you.